So the next speaker will deal with something to do with the brain, I think. Lakshmi Devi from the Mount Sinai School of Medicine is going to talk about big science on a small budget, which I think we just saw too.
Good afternoon, and I'd like to thank Larry for the invitation to talk about a subject that is near and dear to my heart, which is, I modified the title a little bit. It had a small budget, now I made it into modest budget. I think the intent is the same. And then the reason, in fact, this talk is to all of those young and new investigators in the field who are really worried about the trends of funding that we have for drug discovery in the drug discovery process. Whereas the number of new drugs have not increased in the recent years, the amount of funding also has plateaued, especially from the NIH. And what is a NIH academic investigator to do? So I will tell you the journey that I have taken and some of the things that I have learned. Not to panic. Things don't look as bleak as you might think, and it is because of a number of resources that are available to us, and we have to learn to make use of them. So this is a journey, what I have learned from working in being an academic scientist. I started with working with opiates, mainly because morphine is the classic analgesic for the treatment of chronic pain. It also, morphine analogs are used for many other indications, right? So that's great. However, morphine comes with problems. Your continuous treatment of morphine leads to development of tolerance, physical dependence, and addiction, right? So what we really want is a morphine-like compound that has all the good effects of morphine, which is analgesia, without these bad side effects, right? So that is the idea, that's the goal, okay? With a small lab, we're going to do it. Now, morphine binds to a receptor on the membrane, and these are proteins that sit on the membrane, sense morphine binding, and they activate a signal transduction pathway. Since this pathway is activated by proteins that bind to guanine nucleotides, they're called G proteins, not A proteins or B proteins, they're G proteins, and the receptors are called G protein coupled receptors, or GPCRs. These receptors, in fact, are one of the most sought after therapeutic targets for the development of drugs, because there are many different receptors for many different molecules that sense the cell's environment, right? So by targeting these various receptors, the pharmacologists have figured out that there could be selective compounds can be developed for particular receptor target, all right? So this was the status of the field when I entered this field, and we found, in fact, when we took these receptors expressed in cells and looked at what they, actually physically looked at what they looked like, they had a size twice the size of what they should have been, what was predicted to be, okay? And the prediction was that they should be about 50 kilodaltons is the size. But we had 100, 120 kilodalton protein, and that actually worried us and everybody else in the field, because that's not what you expected. The expectation was it should be a single unit, it should be functioning as a single receptor. What we thought we had found was that they actually, two of these units will come together, and that's why we were seeing the higher molecular weight form, that they actually might, if they come together, and they do, in fact, behave differently, they have unique signaling pathway, they might actually function differently. So what did we do? Questioned about 60 years of dogma that these G protein coupled receptors function as single units, activating as a single protein, activating a single transduction pathway, right? So what you need to do when you find data that doesn't fit your hypothesis, go back and question your hypothesis, question the dogma, go back and do some literature search And maybe there'll be, in the publications, there'll be actually information out there that supports your hypothesis, right? So now you redo your hypothesis, rewrite your hypothesis saying that opioid receptors don't only work through single proteins, they might actually complex with each other, and they might have novel pharmacology. So what we did was, in fact, we went and showed that when you take two types of opioid receptors, this is morphine opioid receptor or mu opioid receptor called MOR, and this is a receptor that's related to mu opioid receptor called DOR, and here is from the Lord of the Rings, Mordor, no. This is mu receptor associating with the delta receptor Mordor. So what we had to do is actually to show that they actually come together physically, and we did, by co-immunoprecipitation and pulling them down, and show, in fact, in cells, when you express the two receptors, they come together. We also did bioluminescence resonance energy transfer and fluorescence resonance energy transfer, a lot of jargon, to show that when you express these two cells together, they sit close enough together that light emitted by one can shine the light on the other one. So they were close enough together that they actually were coming together, all in a heterologous system, in a system where we have put the proteins together, right? The question is, are these proteins actually complexing in the brain, right? More importantly, though, if they are complex in the brain, do they change during pathology, a disease? And secondly, if they do change in the pathology, do they behave, they signal differently than the normal receptors, right? If they behaved exactly the same as this or these receptors, then even though they are enhanced in pathology, they have increased levels in pathology, we cannot target it as a drug target, right? So what we needed to do is to actually develop antibodies, reagents that will allow us to specifically localize or visualize those receptors in the brain. So for that, we took an approach that's developed by cancer people that you heard this morning. Someone mentioned about this tolerization. So I'll take you to this approach, which I think is phenomenal. It's really nice. And that is that if you have cancer B and you need to identify a way of recognizing those cells that have cancer B, and it is in a cell that is also has, in the context of other proteins, you want to make antibodies to selective for one type of cancer, right? So basically, you inject the animals with the cells, make them make antibodies, but in fact, kill all of the activated proliferating cells, making antibody to A using a cytotoxic agent, okay? That's called tolerization. And once the system thinks that you don't need to make antibodies anymore to cancer cells A, then you inject the cells that you want the antibodies to be made, right? And then you go back and then check. In fact, you made the antibodies to B, but not to A, and then you characterize, all right? So what we need to do, in our case, we don't want antibodies to mu receptors or the delta receptors, MOR or DOR. We want antibodies to only those receptors that have both in them, right? Mu delta. So we inject the animals with cells that express only one or only the other, right? And then you generate the antibody, kill off all of the activated cells that make anti-MOR or anti-DOR antibody, go back and inject the animals with cells, membranes from cells that have MOR-DOR, make, elicit the antibodies. It actually takes about a year and a half to make these antibodies. And then you go back and actually go back and check, in fact, you made antibodies to this, or the antibody that you wanted to, which is to the MOR-DOR, right? So we went through, this actually took one postdoc about 1.5 years to go through the process, because we went through a few times through the process. Mind you, at that time, it was still dogged that mu and delta didn't come together, okay? So we should not have made antibodies to mu and delta. So this was going in, eyes wide open for failure, right? But at the worst, we would make antibody to mu alone or delta alone if they didn't come together, right? MOR alone or DOR alone. So we went through the process, and we made antibodies and looked in the brain if you actually recognize something that's recognized by the antibody. In the animals that have both mu and delta receptors, more and DOR, you have recognition by the antibody, meaning that these complexes do exist. In animals that don't have mu, more, or don't have DOR, you don't have the recognition. That's good. Now, you've gone through a project, busting the dogma for 2 years, and a postdoc has one figure. Doesn't bode well for, getting jobs, right? So, what you need to do is, okay, you have shown that these complexes exist. But as I said, if these complexes are in fact going to be therapeutic target, they should change during disease, right? So do they actually change during disease? Are they, so it would be a better justification, if you will. So if you're in the opiate field, the disease is addiction. So we drug the animals. This is, you give morphine twice a day in an escalating dose for 5 days. After the 5th day, the animals are tolerant. They need more of a drug to get the same amount of pain relief, and they go through withdrawal, meaning they're dependent on the drug. So you made them addicted. So when you make the animals addicted, you actually find an increase in the relative level of the MORD or the heteromers. We call the interacting complexes heteromers, the heteromeric interactions, right? So then we also went on to show, I'll spare you the detail of all the extra data that we did. We went on to show that, in fact, whereas the mu receptor alone, while it binds to morphine, transduces signal through signal transduction pathway 1, the same morphine, when it binds to the heteromer, the complex, transduces the signal, signal transduction pathway 2. And we showed that this happens by using the antibody that selectively blocks the signal transduction by the heteromer, right? We use that to show, in fact, yes, the signal transduction pathway that's activated by the heteromer is different from that of the homomer. This is a lot of jargon. This is just for those of you who know the field. In the case of mu, there is a G protein-mediated signaling. In the case of mu delta, there is a beta arrestin-mediated signaling. And then we went on to show that this different signaling actually means different things to the cell itself, okay? This signaling causes pain killing, analgesia. This signaling causes tolerance, right? So actually what you need is to block this particular, complex, right? How do you go about blocking the complex? So, to get the drug. So I'm going to summarize here. I want to see where I am right now. Okay, good. So we have gone on to show, in the case of mu and delta, the receptors for heteromers, and in fact, we have gone on to show that we and others have gone on to show in this field, G protein coupled receptors, in fact, do come together. The majority of the time, it is in the case of in different pathologies to form heteromers. The heteromers can be detected in the brain, making heteromer-specific antibodies. We went on to show that in the case of neuropathic pain, when there is chronic pain, and in the case of humans, they suffer from depression. Means there are changes in the brain, even though the pain is in the periphery, there are changes in the brain that some of the times neuropathic pain is treated with antidepressants. So we wondered actually the changes might have been occurring in the brain because of heteromerization, and we showed that in the case of neuropathic pain, there is heteromerization between the marijuana receptor, cannabinoid receptor, and opiate receptor. And the neuropathic pain tends to be a bit resistant to both opiate treatment and cannabinoid treatment. We also went on to show that in the alcohol-induced liver fibrosis, in the liver, there is an upregulation of cannabinoid receptor, angiotensin receptor, heteromers, and the antibodies that we developed are now being humanized for the treatment of liver fibrosis. In this case, in the case of morphine receptors, the complexes are abundant in specific brain regions after morphine treatment, and that they exhibit distinct signaling, so this is heteromers are novel therapeutic targets in ligand screening, right? This is where the problem is, okay, now the academic scientist is done. You identified a therapeutic target, publish the paper, you're done, it's up to the industry, somebody else to do high-throughput screening. Well, I'm a little bit more ambitious than that. So we decided to develop collaborations to identify selective compounds. And you might have heard of NIH's MLCP and library. The NIH actually, but I guess it's like 8-10 years ago, through the blueprint mechanism, had put together a number of screening centers and provided them with the equipment as well as resources for screening for the academic scientists who have identified the therapeutic targets. So what we did was simply to send our assay. Remember, we have an assay to look for heteromer-specific signaling. So we use that assay as a selective assay to screen for compounds. And what they did was to go through the screening that with cells that express our heteromer versus cells that express a non-related receptor, to throw away the ones that are just sticky compounds that would bind to any receptor anyway, right? And from these set of compounds, we rescreened. When they were rescreened, they identified a subset of compounds that were selective for mu delta, but this doesn't actually say that these compounds were only hitting mu and delta complexes, because these cells also have mu alone or delta alone. So we went back and screened, they actually went back and screened those with mu alone, delta alone, and mu delta, and found 94 compounds, and it sent us a list of 94 compounds that are as selective as mu delta, right? 94 is a lot. So we ended up actually taking the top 12 compounds that were more, that are highly selective for mu delta, much more efficacious, and went back and tested if those compounds were in any way better than morphine, in that, did they have analgesia? First important. And if they had analgesia, did they actually develop tolerance and independence? Right? So the analgesia assay is simply to shine a very hot light onto the tail of the mouse, and the mouse or rat will flick the tail because it senses the heat. Except it has morphine on board, it takes a longer time to flick the tail. Okay, so that's what this is. Okay, this is antinociception is simply the amount of time it takes, longer time it takes on board with the drug for it to flick the tail. When we did that, I'm going to show you the data with 4 of the compounds, all were duds, right? We tried to inject them subcutaneously or intrathecally directly into the spinal cord. None of them gave any pain killing, right? Except for one compound, which was resynthesized by chemists, also funded by NIH. Nice, right? They are in La Jolla. And we found, in fact, that actually gave a dose-dependent increase. As you increase the dose, there was more and more pain killing. And most importantly, this pain killing actually came from the activation of the heteromer, because we were able to show this increase in pain killing can be blocked by the antibody, co-administration of the antibody. Okay, that's good. But so this compound is good. We have something that's just as good as morphine. Is it better than morphine? So we did this study where you every day give the drug and see how much pain killing there is, and you find that with morphine, which is the red bar here, by day 8, even though you give the same amount of compound, the amount of pain killing is reduced. This is the tolerance, right? As opposed to our compound, by day 8, there is a slight decrease in the amount of pain killing, but it is not as much as that of morphine. Right? So our compound leads to a lower tolerance compared to morphine. We've actually gone back and shown that, in fact, the compound also leads to decreased withdrawal symptoms. Morphine withdrawal, you might have seen it on TV shows and movies and things like that. It is like tremors and diarrhea and weight loss and chills and all that. Then you can measure that in animals as well, poor things. And in our case, our drug actually causes decreased amount of withdrawal symptoms. I'm not saying that our drug is perfect. It does lead to the development of tolerance, but we believe now that can be modified to make it a better therapeutic to treat pain with reduced side effects, right? So this is a story of how a reasonably funded, or with one NIH grant, an academic scientist actually can make a drug discovery. And I was thrilled. I never thought in my lifetime I'd be able to do this, right? This is the first story. The second story is that of what now, the most recent story, is that of a project that we undertook with no NIH funding, okay? Remember I told you this is G protein couple receptor. So going back to my passions, like you were saying, I started my whole career studying neuropeptides. Neuropeptides are peptide neurotransmitters that help one neuron talk to the other, okay? Neurotransmitters transmit signal very, very quickly. Neuropeptides modulate that transmission, okay? There are many, many neuropeptides in the brain, and we were wondering whether focusing on neuropeptide, can we identify a fundamental problem in the field, right? Fundamental problem in the, and then do literature search and find a gap, develop a testable hypothesis, right? This is coming purely just an idea, right? Start as a graduate student, go back, do the literature search, find something really interesting, come up with a hypothesis, and let's develop it, right? So we focused on, remember, the title of this presentation is Big Science and Modest Budget. So we want to do big science. What is big science? It's a science that makes fundamental contribution to the field. It's a science that will develop a basic technology that's useful to the field. It's a science that's going to reach out, be meaningful to a large population, right? So that kind of science would be the regulation of nowadays, regulation of feeding and body weight, right? So do we know everything about the neuropeptides involved in feeding and body weight? The answer, of course, is no. What you need to know, this is a mouse brain, this here is the nose, here is the back of the brain, and here is the mouse tail here. And there are different parts of the brain that are control different things. We know that already. There's a part of the brain called hypothalamus, and this controls hunger, satiety, and sleep. Okay? And there are a lot of neuropeptides, and so the number of investigators and number of drug companies are very, very interested in looking at what are the neuropeptides that are released by this hypothalamus. And we know that neuropeptides in the hypothalamus translate energy balance to signals, and previous studies have shown that a number of different peptides, the details don't matter. You might have heard of NPY for neuropeptide Y, or AGRP for this peptide, and so on and so on. Some of them make you eat more when they are activated. Some of the systems, when they're activated, make you eat less. So despite these extensive studies, we still don't have a good pharmacological target for the treatment of feeding and body weight. The question is, how are we missing anything? So come up with a hypothesis. Okay, so that's the hypothesis. So how do you look for a new neuropeptide? You can take the brain, mush it up, and then take out all the proteins and look at the peptides. You can do that. Turns out people have done it. Just go back and look in the literature. And this is actually done by Lloyd Fricker's group, who had taken hypothalamus, extracted out the peptides, and put it through something called one of the omics, peptidomics. Is just simply looking at what are the peptides in the brain, in the hypothalamus, right? And you might actually see some of the peptides that you might recognize, some might not, and NPY is somewhere sitting here, and so on and so forth. The most abundant neuropeptide is this guy, called ProSAS, that was named ProSAS by Lloyd, and that is much, much more abundant than anything else. And what is this ProSAS? It is a precursor. What it means is it is made like a protein, and it is processed, which is clipped at certain parts, certain pieces, to give out, make all these little peptides. These are the neuropeptides. So this precursor makes many different neuropeptides. We don't know which one is the active moiety, which is the important one. But the question is, so this is the most abundant peptide. Is it, in fact, in the place where you think it should be? And do they have anything to do with feeding and body weight regulation? So, how do you look at the distribution of the neuropeptide? Well, turns out Allen Brain Atlas actually has posted the distribution of approximately 20,000 genes already ready for you to use. So you go back and look at the distribution of your peptide, which is in our case process. It is present exactly where, in the hypothalamus, where the neuropeptide Y and AGRP are. This is the seat of feeding, right? This is the feeding center of the brain, where our peptide is sitting right where it is, which is called the arcuate nucleus. So this distribution resembles that of the other feeding peptides. The question is, does it actually, any of these peptides have anything to do with feeding? So what we did was to actually inject antibodies to different peptides, and meaning take the peptide away from the brain and see if that affects feeding, right? So basically, you take the animals, put a cannula in, make a hole in the skull, put a tube in, and through that, then you remove the food to make them hungry, and then inject antibodies to the different peptides and see which of the antibodies will cause the animals to eat less. And we found, in fact, antibodies to two of the peptide made them to eat less, right? So we found that this process is one of the highly abundant neuropeptide, is in the center of the regulation of food intake and body weight. Its removal of two of the peptides causes decrease in feeding. So these peptides are likely to activate receptors in the hypothalamus, right? So at the end of the day, okay, we identified in the process-derived peptides as the neuropeptides involved in feeding, right? But what we really want to do really big science, we want to find a small molecule that's going to target these receptors that will stop animals and maybe humans from eating. Or maybe not stop them from eating, stop them from gaining weight, right? So let's go for the gold, right? So where do you look for these the receptors in the hypothalamus? Okay. So it turns out a lot of drug companies have looked for receptors for many of these things and have failed, right? So we thought, I'm not going to do high-throughput screening with all of the orphan receptors. There are a lot of them, and they take a lot of money and effort. So we thought, okay, let's actually take a different approach, use public resources, and turns out that Sean McLaughlin's group had actually taken a piece of the hypothalamus and identified a number of orphan G-protein-coupled receptors. What does that mean is that receptors to which you don't have known ligands for, right? And he had actually made a list of them. So we just basically took that list and then took the top 50, right? 50 is also a lot of things to screen, and I do have a modest budget. So we need to, slim down the list. So that let's actually look at another resource where there might be these receptors. And we have been using these cells, Neuro2A cells, and these are brain cancer cells. They have stem cell-like feature, which means that they have a lot of receptors, and activation or any of the receptors in these cells cause the cells to do different things. Okay, they sometimes grow, they sometimes die, they put out neurites, they put out growth cones and do different things. And I have actually been using these cells to teach high school students about pharmacology. Okay, high school students, when they come to work in the lab, they're not supposed to work with radioactive chemicals and hazardous chemicals or animals or anything. So I just give them just this cell line and teach them how to grow them, and then give them different compounds, different peptides that we had in the lab, and to ask them to throw the peptide and see if you see any phenotype, which means if the compound that you gave them made the cells to do anything. If there is a phenotype, then you can actually understand the mechanism by which this phenotype happened, right? So I'll show you an example here. These are the cells. When you treat, in this case, with one of the peptides, they put out these hair-like things. They're called the neurites. And you can actually put a toxin which specifically will stop this particular type of receptor from working, called protease toxin, that makes them stop putting the neurites, right? This particular experiment told me that these cells actually have the receptor, that is a G-protein coupled receptor for big gland. So basically, we took the list of 50 from the hypothalamic microarray database, matched it with the list of 50 with Neuro2A, purchased 8 cDNAs that can be purchased from cDNA.org, and tested them by expressing it in a cell line where activation, each of the receptor, you express the receptor, put your ligand in, it causes increase in calcium. It's a fluorometric assay, means that you can read it out very easily. Doing that, we actually identified GPR171 as the big Len receptor. But this is good. We still need to go to identifying the compound, right? The clue came where we found that little n, which is shorter peptide here, did not activate the receptor. Big Len did. So that told us that this end bit of the peptide is in fact the business end that's involved in activating the receptor. So to go get at the small molecule that is able to activate or inhibit the receptor activity, we developed collaborations, and we have an experimental therapeutic institute where what they do is they take the structure, the amino acid sequence of the proteins, and build a homology model based on the crystal structure of other G protein coupled receptors. Okay? So basically, they visualized how this receptor should be looking at, and in fact, virtually docked the small peptide into the receptor, identified the binding pocket, and then went with a small molecule library of compounds and to see which of these compounds could bind to the receptor the way the ligand binds, endogenous ligand, the peptide binds. Right? By doing that, they gave us 30 compounds. We tested them. One of them caused an activation. 4 of them actually blocked the activation by the peptide. And so, cut to the chase, the small molecule compound that actually blocks the activity of the receptor is in fact able to decrease feeding when given chronically and decrease much better the body weight of the animals. Right? These are in the in this first week, second week, and third week. Right? So, hypothalamic G protein coupled receptor is identified as a big Len receptor, and using homology modeling and virtual docking, so this is not by doing high throughput screening at all. This is just by simply using the existing information about the structure of the compounds as well as the structure of the crystal structure of the receptors. We were able to predict 30 compounds would bind. We went back and tested these 30, identified 4 compounds, out of which an agonist causes an increase and an antagonist causes a decrease in body weight. So what we are doing now is, with the chemists, actually trying to improve once again the pharmacokinetic properties, meaning that making it into a drug rather than from a hit to the lead, so that it is now being, it could be a better therapeutic for the treatment of disorders. So these are the people. This is actually a view from my lab. I'm from Manhattan, I told you, right? This is Central Park. These are the people actually. It's a small lab that did all the work. Achla Gupta is the one that made all of the antibodies. Yvonne did the earlier characterization of the pharmacology, and these are the people now doing behavioral studies. And we have had help from a number of groups. Tibor Harkney from Sweden did the histochemistry. Peter Harder is the screening center in La Jolla that did the high throughput screening. Ed Roberts is the chemist who's optimizing the compounds. Mihai is the molecular modeler, and Lloyd Fricker for all of the studies, and help with reduce the number of slides. Thanks.
I have an easy question, at least I think it's easy, regarding neuropsychiatry and the body's ability to deal with pain by producing endorphins. For instance, when I was a kid, back during the Vietnam War, I busted my arm really bad. By the time I got to the Naval Hospital, I wasn't in pain anymore because my body had produced those endorphins. And I was just wondering if you could kind of explain to us a little bit about that, the body's natural ability to do that. And just to quantify that, the Colorado State Legislature has sent to the governor's office a bill licensing naturopaths, and a lot of people think naturopaths are kind of like pseudoscience, but it's biochemistry and 4-year degree, and I guess Linus Pauling is one of the founders of that kind of teaching, and make it quick, he coined the term orthomolecular. In other words, before you use drugs, which again you noted can have side effects and can actually inhibit the body's ability to heal, you look at the biochemistry of an individual, and taken in a whole, people with psychiatric conditions, for instance, you change their diet, and then you need a very little amount of a psychiatric medication to actually treat that condition. And I know, again, Linus is kind of criticized, but I kind of believe in someone that's got 40 plus PhDs than someone who only has one PhD. So anyways, that's my take. Thank you.
Right. So the question actually comes down to, if we have endogenous morphine-like compounds, could we in fact simply use those to kill pain? That we shouldn't be developing tolerance and dependence. And turns out in high concentrations, and this was actually tried, endorphins, which are the ones that give you runner's high. In fact, the body will secrete a lot of these endogenous morphine-like compounds, and now we know that there are endogenous marijuana-like compounds that the body can also secrete. They're called endocannabinoids, and these compounds do work through the pain-killing receptors. However, a study that was carried out actually in China for the terminally ill cancer patients with the pelvic pain were administered endorphins, and they did develop tolerance. Right. Having said that, most of the studies nowadays have support that acupuncture analgesia is through the release of endorphins, endogenous endorphin-like molecules. And this was done, in fact, in animal models where it's called electroacupuncture analgesia, where you can actually take their paw and give them the mild electric acupressure, if you will, and then the tail flick will be, delayed. So you know that they're releasing the endogenous cannabinoid and endorphins, right? But if you in fact put antibodies to endorphins into the spinal cord, then the pain killing is gone, meaning that they sense the pain. So you know that the endorphins are released, and that's what they're causing during the electric shock. So finally, the professor from Beijing who developed this, Dr. Zhisheng Han, made a machine where you could put your hand in and you could stimulate the endorphin release, right? And use this for the treatment of narcotic addicts in jail. So it's called Han's machine, and I've never been able to test it myself by saying how high I'm going to feel. But apparently, this release of an endogenous that's a treatment. Methadone treatment is a treatment for drug addiction, right? Opiate addiction, which is you give small doses of orally active opiate so that the addicts wouldn't go through the highs and lows of drug addiction. Instead of taking an external opiate, you release your endogenous opiate through this electro acupuncture, if you will.
Okay, Amy.
So thank you very much. As a young scientist, we're very much so looking at how to do big science on a modest budget. And so one of the things that I was wondering about is when you go out to make the collaborations for the things that you can't do in your lab, is that something that you do through your network, or is that something someone has a paper, they've done this great work, and you just email them and say, hey, I'd really like to work with you? What do you think is the best way to go about it?
Well, the best way probably to go about it is to come to conferences like this and others and just to develop a network rather than just emailing someone. What has worked for me the best is actually to share with them my data, and you have to be a little bit masochistic for that because they can take your data and they run with it. But share the data and say, like, this is what I have, and this is where I think your contribution would work very well. We'll be, you have to, for collaborations, you have to be upfront in it's what you're going to get and what we're going to get, and your contribution will be recognized based on how much contribution you do. You'll be co-authors, where the author should be. All of that should be upfront discussed. And so the collaborations for me have been mostly through, and it becomes easier when you sit down and have a cup of coffee or have lunch and then talk and discuss and all that.
I wonder if Matt has something to say about that, if he's still awake over there. Since you proposed the opposite thing, which is just throw it out and there's all these strangers. And actually, I was going to ask you this. Didn't you have a bunch of nuts? How many crazy people?
No, you don't. I mean, you get people who are enthusiastic, but you get...
Oh, enthusiastic nuts, you mean?
You get nerds, you know, get nuts. No, so I mean, yeah, I mean, what I was thinking about that was that, yes, you can, if you put data into the public domain and if you publicize it, which is not a natural feeling for us because we publicize work we've finished, usually. You don't publicize work you're doing. But if you do that, then you can get people you don't know coming to contribute to projects, yes. I would add that.
But along those lines, if you're studying morphine addiction, it's a totally different issue as far as the public. It's like me saying, oh, vaccines are good for you, right? There's 30,000 people out there going to tell it causes autism, blah, blah, blah. So you're going to have to, there's going to be more nutty people is what I mean.
Your nutty people is my outreach.
I was going to ask you about,
Oh, yeah.
Just to be fair, though, I mean, the reliance on the literature for finding stuff, given that the literature is ever expanding and pretty much anything gets published, there's a great deal of nuttiness within
Right. You have
so-called
to be,
science.
yeah, you have to be thorough about it, and you actually have to evaluate. So, do the literature search. What I mean by that, actually, I give this similar talk about the discovery process. What's involved in the discovery process, right? Most of the time, when the students come in and they're given a project based on the foundation that's set by someone else, that's being picked up by the PI, they actually have to go back and what were the fundamental papers that set that foundation. If that foundation is not strong enough, then all they're building on is house of cards. So that's where the literature, go back to literature search. But you have to be thorough as to go to the original literature and see under what context that truth is the truth.
I wanted to ask you a question about the dimerization issue.
Yeah.
How promiscuous are G-coupled protein
couplers.
receptors in this regard? For example, in the lung, there's all kinds of issues about asthma and these kinds of receptors. And do you think there also, for example, in the asthmatic lung, there might be abnormal dimers being formed, or is it just neurological?
No, not at all, not at all. We actually published a paper very recently that there is dimerization between CB2 cannabinoid receptors and CXCR4 in the T cells.
Oh.
It's human T cells.
I'm sorry to hear that.
The dimers. So we did a number of studies to actually address that question, and we find that there are proteins normally that keep the dimers from forming. And then the conditions of disease, when that protein that keeps them apart is not there, the dimers come together. And then there are proteins that actually degrade dimers. And when they are not there, then you have enhanced dimers. So there are proteins that are chaperones that will facilitate dimerization or keep them away from being dimers.
Over here.
Yeah.
Interesting about how science in my infancy seemed to be done, and the researchers using what was in the public domain. I have one question, is curiosity. When you've created the new analgesic, do you protect against addiction by getting rid of tolerance? And that's just a curiosity. My real question is, you've now put in the public domain in this talk really cool information, not only about your process, but about your product. How do you protect yourself, given the previous conversations we've had about academia's paranoia, possibly about open sourcing, et cetera, from somebody coming in saying, I'm going to take that G protein receptor now, and I'm going to create the next anti-obesity drug, and I have $1 billion to do it. This looks promising.
Right. So your first question first, which is our compound has the ability to develop addiction. It is not as bad as morphine, but there is a little bit of the ability to develop, the animals do develop dependence. And now we know why, because this compound is not as clean as it can be. It does hit more alone, receptors alone. So that has to be, changed, modified to now only hit the complex and move along. And the chemists are doing that. And if you noticed, I didn't show you any structures. And I didn't even give you the numbers, right? I called it MS there, right? But to be fair, all the information that comes from NIH screening is in public domain. It was put in, I think like the week after it was screened. All of that data is out there. So that was kind of the downside of all of using the NIH library screens. But for me, it was the process. I'm not in it for the money, sorry. I
Okay,
think
we'll be
there's
back at 4.
one last
oh,
question,
sorry,
maybe.
one question.
Yeah, shout it out.
And what he said to me was that if you want to keep something a secret, then you should publish it, because nobody reads the literature. He said that if you want to make sure that everybody knows about it, then you should tell your best friend and tell them not to tell anybody.
With that,
Okay,
with that.
thank you very much.